Okay, welcome to the review for immunity. I'm going to walk through some of the main components of immunity that you'll be at least tested on in class. The AP, obviously, you should keep reviewing this and making sure you understand how it works because they're going to expect that you understand it in pretty good detail. So starting off with immunity, this is our ability to fight off diseases and fix problems possibly in our bodies. The first piece is a non-specific component. Non-specific meaning it really doesn't matter what's coming into our body. We're not happy with what the situation is. So it could be a bacteria, it could be a virus, it could be a parasite, it could be a foreign organ. Our body doesn't recognize it, so it's going to try to destroy it. So non-specific, we have two lines of defense for non-specific. The first line is your skin, your mucus. I think I spelled that wrong. And enzymes that you secrete to the surface of your skin, like lysozyme. Those things are actually pretty good, but as this image is showing right above us, this is can be breached, obviously, if you end up getting cut or poked or something along those lines. And if that happens, then we move into the body, the bacteria that might be on, for example, this little needle here, now is inside and has passed our epidermal and dermal layer of skin. And now we're in that interstitial area between the dermis layer and possibly into some muscle tissue or near some layers, like this is a blood vessel here. And now the bacteria is inside. So the second line then is going to be the attack of white blood cells, right? So you have macrophages, as you're seeing here, a phagocytic cell that's going to try to start eating some of these bacteria. You also have the eosinophils, which are white blood cells that are going to start releasing histamines, right? And histamines allow for vasodilation, meaning that the blood cells start dilating. We're going to send more blood to the site. It starts getting inflamed. And the reason that that's good is when you do that, you're going to be sending more platelets, which is going to allow for clotting if we have a surface opening. And it's also going to send more white blood cells to the site to start fighting that infection that might come in because of that initial penetration of the skin and mucus enzyme layer. So we get that initial attack of the white blood cells. That first phagocytic cell that starts eating is a macrophage, and that's going to actually kick off our specific defense, right? So when we start kicking in specific, here's the second image here. We start kicking in the specific after that macrophage starts presenting pieces of the antigens. The antigen is anything foreign into your cell, and that first macrophage is an antigen presenting cell, an APC. And the way that it presents it is via an MHC, a major histocompatibility marker. Those are the yellow pieces on top of the macrophage here. And as you can see, it's presenting a little teeny fragment of the antigen. So see how the antigen has these little red dots? The MHC is presenting a little red dot. So that is going to be, and this is the MHC2. We'll talk about MHC1s. Those are found on normal body cells. So this macrophage presents that little piece, and I like to think of them as flags. They're basically surface carbohydrates, and normally the flag is a nice, happy version of you, right? But if we end up eating something that's wrong in our body, as this macrophage is doing, then it's going to be presenting a picture of the virus or the bacteria, right? And now we have an image basically for the system to start learning. So this macrophage then starts secreting these little dots, right? And those little dots are a chemical. It's called interleukin-1. And interleukin-1 calls in the helper T cell. As you can see, the helper T cell has a piece here that's going to bind in with that MHC1 or MHC2, and it's going to read it. And then it starts secreting another chemical which is called interleukin 2. And interleukin 2 is going to start calling in our specific immune system. We're going to specifically fight that antigen that has entered our body that we know is there, and in turn we start differentiating, right? So as the image is showing, we're going to do some mitotic divisions. We're going to call in B cells, and we're going to call in, it doesn't show here, but we're going to call in T cells as well. So at the same time that we make these B cells and T cells, we're also going to make memory cells of both of them. So you're going to make memory B cells, you're going to make some memory T cells, so that if we get a second infection of this antigen, we can fight it much faster. All right, so we start making B cells and T cells. Now we need to talk about where those B cells and T cells are made, and blood cells in general. So blood cells in general are going to be made in the bone marrow. Their stem cells are called adult stem cells that are found in the bone marrow, and we're going to start making red blood cells there and all the various white blood cells. So the ones that we're focusing on right now are the B cells and the T cells. So where the B and the T come from is the maturation component. So B cells are going to mature in the bone marrow where they were originally started. And then T cells migrate via the blood system up to the thymus, which is in your chest area, right around your clavicle, which is the bone that's right across the front there at the top of your rib cage. And that thymus is going to mature those stem cells into T cells, all right? So they can be T helper cells, or they can be killer T cells or cytotoxic T cells. So now we've called in B cells and T cells that are going to specifically look for this antigen that has now infected your cell. The B cells, as you can see here in this image, mature into plasma cells, and those plasma cells are going to start secreting these things called antibodies. Okay. Now, antibodies are great for two purposes. One, they're going to start clumping up the antigen that is in your body. So if it's a bacteria, it's going to take bacteria and it's going to start sticking bacteria together. And why is this advantageous? Well, instead of the macrophage eating one at a time, now this macrophage can come in and consume 4, 5, 6, 7 as a clump. So that's going to help. That's going to speed up how fast we consume the bacteria. But also the antibody has a joint system with some proteins, and that can actually punch holes in bacteria's cell wall and plasma membrane, and it'll lyse the bacteria. So antibodies serve as a dual function. All right. And by the way, this is called humoral immunity. Humoral. Wow. I think my spelling is way off this morning, but if it's right, good for me. If it's wrong, don't judge me. All right, so humoral immunity, we're making B cells. The other side is called cell-mediated. And cell-mediated, we're looking at T cells, specifically killer T cells or cytotoxic T cells. As this image kind of shows, cytotoxic T cells are going to look at virus-infected cells, bacterium-infected cells, and cancer cells. Now, these are your cells, right? Your body cells that have been infected. So what the killer T cell is kind of the idea of amputation. So if you have a damaged tissue that no longer functions, it's best to cut it off, otherwise the infection can grow and it can start spreading throughout your body. So what the killer T cells do is they recognize cells, so this would be your cell, and your cells have flags normally as well. So this flag is, any flag is an MHC. This is the MHC1, and this flag will present not a happy picture of you, but a picture of the virus or the bacteria or an abnormally dividing cell, right, as in cancer cell. And that flag will help the killer T cell recognize which cells have issues. And so then the killer T cell binds up with that cell and starts, as this gun is showing, shooting holes in the virus-infected cell or the bacterium-infected cell or the cancer cell, and punches holes in them and they explode. And it's called controlled apoptosis. Apoptosis meaning we're destroying the cells. So we're going to do that controlled apoptosis with killer T cells, and that's cell-mediated. And you think to yourself, well, why does cancer even persist if this is a way for us to fix the issue? Well, yes, it is. We can stave off a lot of things, and I'm sure there's times in our bodies that cancer starts showing up, and we don't even show signs of the cancer because we have this immune system that fixes it. But if it goes too fast or too haywire, our body can't keep up, and the cancer will spread. All right, so that's the connection as far as cancer is concerned. So that is specific immunity right there. If the B cells and the T cells are involved, that's specific immunity. Make sure you understand the difference between humoral and cell-mediated. All right, that being said, we move on then to how primary exposure looks versus secondary exposure. So this graph is straight out of your book, and it's showing antibody concentration. Once again, antibodies made by B cells that mature into plasma cells. So the antibody concentration is showing you how fast the body is responding to a certain infection. So we expose here, right, and it takes, as you can see, almost a week before antibodies start kicking in. And then by the end here, right, we're near the end of the infection, so we're at possibly 3 weeks or so. The bacteria is pretty much killed off, or the virus is killed off, and our body is starting to recover. So because of the duration of time and how long it takes for the antibodies to build up, we end up getting a sick feeling, whatever those symptoms are of that bacteria, virus, or tapeworm, or fluke, or whatever it is that is inside your body. It takes a little while for your body to right itself. Now, second exposure, remember that we created those memory cells, memory T cells, both helper and killer, and memory B cells. If we get exposed to that same exact antigen again, maybe a week or 2 or 3 weeks or 4 weeks or something along the line later, well, then we can kick in much faster. We don't have to do all the passing of signals. We actually kick those memory T cells and memory B cells into action fast, and in turn you can see the spike of antibodies goes through the roof within a day or two. We have a pretty steep slope, and in turn we knock down that bacteria or virus very quickly, and we virtually feel no ill symptoms whatsoever. All right, so as you can see, if we have any exposure to a second antigen, well, that's a whole new organism or a whole new piece that our body doesn't recognize, so we have to go through that whole process again, and we'll feel ill symptoms. So how does this connect to vaccinations? Well, vaccination is a weakened form of the virus, or a damaged virus, or a piece of the virus. So what you're doing is you're exposing your body to the antigen without the possibility of the antigen spreading. So you're putting up yourself through specific immunity, and it's active immunity because your body runs through it, and in turn you develop the ability to make memory cells, so that if you actually find an active form of the virus, or if we're doing a bacterial one, your body will fight it off faster. So you question, well, why is it that we have to get a vaccination, for example, influenza every single year? If it's one organism, does it make sense that it should last for our entire life? Yeah, but as we already know, organisms change over time. That's evolution. And so in turn, the surface antigens change every single year. That virus mutates. And because of that, we have to get a new flu vaccination every single year. And same thing with bacteria. Even though you might get different types of bacterial infections, you can get them again because that bacteria has mutated or changed over time. So that's active immunity if you actually fight off the disease, or you're getting exposure to the disease at one way, shape, or form, and your immune system has to go through it. Passive immunity is if you are getting some sort of immune response without having to work through it. So passive immunity is mostly mother to child. And this happens when the baby is in the womb. So the mother maybe gets sick, or the mother has presented or made antibodies. They're going to pass them because they share blood with the child. And in turn, the child gets some of those antibodies, and it continues on as the mother breastfeeds. So if the mother breastfeeds, she also gives antibodies to the child, and the child gets better immunity because of it. So there's a health benefit in breastfeeding. The other way that a person can get passive immunity is if they do a blood transfusion and there's antibodies in the blood, they can also get some of those antibodies, and in turn have a better immune system. Last piece connected to this is our virus discussion. So this is showing you HIV. And HIV is kind of a nasty little booger because it has, as we already talked about, it has the ability to basically mess up your entire system and shut it down. All right, the HIV virus will cause the immune system to cut out. So the way that it works is it uses these surface glycoproteins, as you can see in the picture, to dock up with your cell, and then it injects its components inside your cell, specifically RNA, and then this enzyme called RNA reverse transcriptase, which we've already talked about. And in turn, it turns its DNA, its RNA into DNA inside your body, integrates it into your genome, and then your cells start becoming manufacturers of HIV. When it gets really bad is when it starts attacking your helper T cells. So what happens is it starts killing off your helper T cells, and since your helper T cells is the dividing row between passive and active immunity, your active immune system pretty much shuts down. And if that happens, then you have AIDS. So HIV and AIDS are two separate things. AIDS is basically HIV has destroyed your immune system so poorly, or so much that now you basically can't fight off any infections that you might get. So people with AIDS don't die because of the HIV directly. It's more indirectly because it's pneumonia and strep throat and things like that that end up killing the person that has AIDS. Okay? And this concludes our immune system discussion. If you have any questions, by all means, email or talk to your teacher. Wish you the best of luck.